I'd like to do today is, first of all, give you a brief introduction to some of the work that we do here in the lab. And then I'm going to talk specifically about one technique that we use called H reflexes, and how we use it and why we use it. And then, so that I'm hoping will take about 20 minutes. And then at the end, so we are actually in the lab right now. I don't know if you can see, but we're actually in my lab. So at the end, Mateos and I will demonstrate how we produce H reflexes in the lab and some of the technical aspects of how we do that. So first of all, I'd just like to give a brief introduction to some of the research that we do. Please let me know if the slides are moving along. So I just changed the slide. Hopefully that worked at your end. So a lot of the work that we do in the lab has to do with electrical stimulation of muscle, which is also called neuromuscular electrical stimulation. And so one of the main uses for electrical stimulation of muscle is for people who've got injuries to the nervous system, so they can't voluntarily contract. So we can generate contractions for them to help them move, for example, to exercise and ride a bike, but also to improve the quality of the muscle, so to reduce muscle wasting. Electrical stimulation can also be used to strengthen pathways between the brain and muscle for people who have maybe an incomplete spinal cord injury or a stroke. But I'm not going to talk much about that part of our work today. So about 12 years ago, when I was doing my postdoc in Sydney, we accidentally made this discovery. And so what we found is shown on this slide here. So we were electrically stimulating muscles, and we were stimulating them in two different ways, and that's what's shown on this slide. So one way we stimulated it was at a low frequency and using a very narrow stimulus pulse. And what you can see on this slide by the thin line is that that generated approximately 5% of a maximum contraction. But then what we found, and this was not what we expected, was when we stimulated at a higher frequency, so more pulses every second, and each one of those pulses was a longer duration. So instead of 0.05 milliseconds, it was 1 millisecond. And what we found is we got a lot more force out of the muscle, and you can see that in the dark trace there. So we weren't expecting this, and so our first question was, where is this extra contraction coming from? And you can see in this figure it's generating about 3 times more force than when we stimulated it the other way. So we call this extra force or extra torque. And we call it WPS, or wide pulse stimulation, because we're using that 1 millisecond pulse width compared to the really much narrower 0.05 millisecond pulse width. So we predicted, or we hypothesized, that extra contraction was coming from pathways that travel through the central nervous system, or basically reflex pathways that go through the spinal cord. So the first thing we wanted to do was test that hypothesis. Does this extra contraction come from the central nervous system? So we delivered the electrical stimulation. Before and after, we blocked the nerve. So if you look at this figure here, you can see the muscle, you can see the stimulating electrodes there, shown by those horizontal lines, and you can see the spinal cord shown there, and a reflex pathway through the spinal cord. So what we did is we blocked the nerve. We put an anesthetic on the nerve between the nervous system and the stimulating electrodes. And we delivered the stimulation before and after we did that nerve block. So what you can see on this figure is the contractions, or the force of the contractions before we did the nerve block. And you can see we got fairly large contractions, about 15 to 20% of a maximum voluntary contraction. And then we block the nerve. So with the nerve, before the nerve block, reflex pathways can contribute to the contraction. But basically what the nerve block does is it disconnects the muscle and the stimulation from the central nervous system. So anything that we see, any part of the contraction that we see during the nerve block cannot travel through the central nervous system. And what you can see here is during the nerve block we got much less force out of the muscle than before the nerve block. And so what that showed us is that all of that extra contraction is coming from those pathways through the central nervous system. So either through the spinal cord or pathways up to the brain and back down to the muscle. How are we doing? Is that clear so far, or am I going too fast? Because I know I speak quickly for I discovered that when I was in Brazil. So please tell me if you have any questions or if I'm going too fast. So the nerve block data said something is happening in the nervous system that's contributing to these contractions. The next thing we wanted to look at was, do H reflexes contribute? And I'm going to talk a little bit more about H reflexes in a moment, but basically H reflexes travel through reflex pathways through the spinal cord and back to the muscle. And so what we did is we actually recorded from the muscle when we were delivering the electrical stimulation, and that's what you can see here. So the top of this figure shows the force that we got out of the muscle when we delivered the stimulation at 20 hertz, and then at 100 hertz, so at a higher frequency, and then at a lower frequency. And the bottom of this figure, you can see the EMG. And if you look at the left side of that EMG, you can see a very large H reflex. And then after the 100 hertz stimulation, you can see those H reflexes come back. And so this suggested to us that transmission along that reflex pathway through the spinal cord is contributing to the contraction. And this is just summarizing that the you can see the very large first H reflex, that first dot to the left, and then the H reflexes get smaller, and then after that 100 hertz stimulation, the H reflexes are bigger, and we think that it's those bigger H reflexes that are generating that extra force. So the H reflexes were larger after that high frequency stimulation, and we think they contribute to that extra contraction. So just to kind of summarize what we're thinking about how electrical stimulation generates contractions, it generates it by two pathways. One is by stimulating motor axons right underneath the stimulating electrodes, and that generates a contraction through a peripheral pathway. And the other is underneath the stimulating electrodes, we activate sensory axons, and that goes into the spinal cord by this H reflex pathway and contributes to the contraction that way. So currently we think that it's a good idea to try to produce contractions through that reflex pathway for rehabilitation, because it's a more natural way to produce the contraction. And some of the other questions are actually probably I'm not going to talk too much about those current research questions right now. I want to focus on that H reflex and how that contributes to the contraction. So one of the reasons that we think it's a good idea to produce contractions through that pathway for rehabilitation is because when you generate contractions through reflex pathways, it recruits those motor neurons in a physiological way. And what I mean by that is it recruits motor neurons according to Henneman's size principle. So what you get is the most fatigue-resistant motor units first. And one of the problems with using electrical stimulation for rehabilitation, so for people with a spinal cord injury, is that the contractions fatigue really quickly. So basically the contractions get smaller and smaller and smaller very quickly. But if we can produce these contractions through the spinal cord, through that reflex pathway, we can recruit the most fatigue-resistant motor units first, and we think that that is going to improve the way that we use electrical stimulation for rehabilitation. And we think it's going to produce contractions that are more fatigue-resistant. And fairly recently we have some data that supports that idea. Felipe, can we load up the other PowerPoint presentation, the H-reflex one?
Yes, I will do it right now.
Okay, thank you. So what I'm going to talk to you now is a little bit specifically about H-reflexes. So I'm going to just go over a few slides about H-reflexes. And then Mateos and I will hook me up and we'll show you how we generate our H-reflexes in the lab. So the muscle that we most commonly use is the soleus muscle. That's at the back of the lower leg. And the reason really for that is just it's easiest to produce H-reflexes in that muscle compared to most other muscles. But you can generate H-reflexes in most muscles. So reasons why you might want to produce H-reflexes, we are using it more as a way to generate contractions, but a lot of people use it as a way to test the excitability of the spinal cord. So because you're sending in a big sensory signal into the spinal cord and you're activating motor neurons and it comes back to the muscle, you can get a measure of how excitable those motor neurons are. And so that's one reason that people use the H-reflex technique, is to test how excitable motor neurons are when you do different tasks. So for example, some of the early work on H-reflexes was looking at how they change when you go from standing to walking. And so H-reflexes are very large when you're standing, and they get much smaller when you start walking. And so the excitability of that reflex pathway gets turned down by the nervous system when you go from standing to walking. And so that's an example of how the nervous system controls transmission along reflex pathways during different tasks. And so we're just trying to load up a few slides that I have about H-reflexes, and it looks like they're coming on right now. And so I just wanted to talk a little bit more about H-reflexes. There it is right there. So again, you see this is the similar diagram that I showed you before. So you have the muscle on the left, you have the spinal cord on the right, and you have your stimulating electrodes in the middle there. Yep. Oh, can you guys see that mouse? Maybe you can. So there's the muscle, there's the spinal cord, and there's your stimulating electrode. So when you stimulate a peripheral nerve, you get two responses. And you get two responses because you're stimulating the motor axons under the electrodes and also the sensory axons under the electrodes. Oh, and it's not showing up for some reason. We'll show you some M waves and the H reflexes on the oscilloscope in a moment. But the M waves happen about 5 or 10 milliseconds after the stimulation, and they're from stimulating the motor axons. The H reflex takes longer because it travels a longer pathway. It goes through the spinal cord, so the H reflex travels pathway through the spinal cord and back down to the muscle. So that takes longer. It happens at around 30 or 40 milliseconds. And so it's the size of that H reflex. And I think we just lost our presentation here. Hopefully it's coming back. It's the size of that H reflex that gives you an idea about the excitability of the spinal cord. Oh, that's the PDF?
Yeah.
Okay. Not sure how well this is going to work. Okay, so when you stimulate that peripheral nerve and you slowly increase the intensity that you stimulate at, you recruit more and more axons, and the M wave gets progressively larger. But the H reflex gets larger for a certain period of time, and then it gets smaller again. So I wanted to talk a little bit about why that is. And so that is shown on this slide here. Oops. There we go, right there. So if you go, if you look at the right side of this figure and you go from the top to the bottom, that is showing the EMG responses as you slowly increase the stimulation intensity. So the first line is a flat line. There's no response because you're not stimulating anything. And then as you slowly increase the stimulation intensity, you see M waves get bigger, and the H reflex gets bigger, and then it gets smaller again. And the reason for that is because when you stimulate those motor axons, signals travel towards the muscle, but they also travel backwards towards the spinal cord. So what happens is the signals are traveling backwards up the spinal cord, but signals are also traveling from the spinal cord down. Whoops. And they collide in those motor axons, and they can't get through. So the H reflex gets smaller and smaller and smaller as the M wave gets bigger. And so what that does is it produces, so you can see on this panel, as you go down, the M waves get progressively bigger, the H reflex gets bigger, and it gets smaller. And the reason it gets smaller is because of that collision in the motor axons. And that is, I try to show that here. So in this figure, it's a little bit, there's a lot of detail. But if you start, and I hope you can see the mouse here. If you start looking at this panel here, the one that says threshold H, that's an example of a very low stimulation intensity. And you recruit the smallest motor neuron in the spinal cord, and you generate a very small H reflex. If you go over to the next one on the right, it says increasing stimulation. You turn up the stimulation, you get more motor neurons contributing to the H, and you start getting motor neurons contributing to the M wave. And then if you go right down to the last one that says maximal stimulation, you see all these axons that are in yellow. All of those axons are being blocked. So what that means is the H reflex can't get through. So in this particular situation, you get a maximal or a large M wave and no H reflex. And so that's shown here, and this is the last slide that I want to show you. On the left-hand side, you can see the EMG responses. So the first one's a flat line. There's no response. And you see a small H reflex. And then you see this M wave that gets bigger and bigger and bigger.
Yeah. Click on the screen. Exactly where it is.
Click.
Yeah.
Oh, look at that arrow. Ah, beautiful. Now they tell me. And then you see this H reflex getting smaller and smaller. And that's because of that collision in those motor axons. And here we have what's called a recruitment curve. So shown here is the M wave curve, where we have stimulation intensity along the x-axis and response amplitude on the y-axis. You can see the M wave just gets bigger and bigger and bigger. The H reflex here gets bigger for a certain period of time, and then it gets smaller, and eventually it goes back down to zero because of those collision in the motor axons. So what we're doing in the lab is we're trying to generate contractions through this H reflex pathway in order to produce contractions that are more fatigue resistant. So what we'd like to do now then is we're going to go, and I'm going to hook myself up to the equipment, and Mateos is going to demonstrate how we produce H reflexes in the lab. So that's the end of the PowerPoint thing. You can take that off if you want, or you can leave it there, whatever works. So what we're going to do is we're going to stimulate my soleus muscle. So that's on the back of my leg here. So I've got electrodes here in the back of my knee. Thank you very much. So we're going to stimulate the tibial nerve here, and we're going to record from the soleus muscle down here. And we probably want to have that looking at the screen.
Yeah.
So just give us a minute while I hook myself up. So these are the stimulating leads. I'm going to hook them up to the stimulating electrodes here in the back of my knee. And I'm going to hook up the EMG electrodes to record from my soleus muscle. Okay, let's see if I can take it away.
What you want to do manually? You want to see curve?
Oh, let's just first of all see some waveforms. Oh, okay, we're getting stimulated. So I'm going to turn this around so you can see the computer screen that we collect our data on. There we go.
Sorry, too many cables.
Okay, there you go. Beautiful. Can you see? Oh, I don't know if they can see that.
Philippe has I think they can see. Philippe has a chat here. Just a second. Can you guys see it? The screen? Felipe? Felipe?
Yes, it's working. Yes.
Okay. Thank you. Thank you. Yeah. You can
Okay.
see it. Yeah.
So I guess let's kind of do a recruitment curve, or at
Okay.
least show some M's and H's.
Okay. You can chat. Feel good to hear you.
Yeah.
Let's see. Are you getting pulses?
I think it's delivering and you're delivering at the same time.
Oh.
Yeah, there's one.
Freeze the screen.
Or just take it off. Just do it manually, can you?
That's what I'm trying to do. I'll put a very long pause.
Yeah, that's good.
Like 20 seconds. Okay. Okay. So we are stimulating Dave right now.
Can you change the? Is this okay this way or no?
What do you mean?
Zoom this way.
Oh, the zoom, I think I can change that.
Okay, that's cool.
So every time that I stimulate, this part here will change. Okay, this big wave here means an artifact. That means I stimulated. Okay, so everything that is important will happen after the M wave, after the artifact. So this is a very low stimulation intensity. You cannot see really anything. If I increase a bit, you'll see a huge curve showing up here. And this is what Dave was talking about, the age reflexes.
It's huge.
So, Dave has a beautiful central nervous system. He is very proud of it. Sorry.
That's okay.
Okay. So this little thing here means the stimulation artifact when I stimulate it. This small curve that I hope you guys can see is an M wave, and this big curve here is an age reflex.
Yep. So what we're going to do now. So remember, the M wave is from stimulating motor axons. So that's why it happens immediately after the stimulation artifact, because the pathway is very short. It only has to travel from the back of my knee down to the muscle. The H reflex takes about 35 milliseconds, and that's because the pathway goes from the back of my knee up to the spinal cord and back down again, and activates motor neurons in the spinal cord. So what we're going to do now is we're going to slowly increase the stimulation intensity. And so what you're going to see is the M wave get bigger, and the H reflex eventually will start getting smaller. And that's because of that collision in those motor axons.
So I increased a bit the stimulation intensity. The H reflex got bigger, no change on the M wave. Increased a bit more, stimulated again.
There you can see the M got a little bigger.
I hope you guys can see that this first wave here got a bit bigger. Then I increased again.
Whoa!
This one got way bigger. I put it a bit lower just to see the increase. So this one got a bit smaller. I reduced the stimulation amplitude a bit. So if I increase a bit again, it'll get bigger. They're both the same size now. If I increase a bit again,
Yeah.
this one got bigger and this one started to get smaller. I increase a bit again. I'll just change the scale here. So this one I'll increase again the stimulation amplitude. It's got bigger, smaller. Increased again, stimulated. Increased a bit, it's very tiny. Increased again, stimulated. They're starting to, this one is starting to disappear. I increased again. And again. And
Do it again.
I stimulate again. And I hope you can see that the H reflex completely disappeared and we only have an M wave here. So this is a very high stimulation amplitude, and we are not getting any H reflexes.
And so now what's happening, so we would call this M max. So if you notice, Mateos can turn the stimulation up again.
Increased, stimulated.
And the M wave doesn't get any bigger. And that's because we're stimulating every motor axon. So typically in our experiments, we always record M max, and we use that to normalize our H reflexes. So we always report our H reflex amplitude as a % of M max. And that gives us a bit of an idea about how many of the motor neurons to that muscle that the H reflex is recruiting. So for example here, this M wave is about 9 squares. You probably can't see the squares, but anyways, it's 9 squares high. And then, so what we're going to do now is we're going to turn the stimulation down and see what the biggest H reflex we can get. And typically, the biggest H reflex you can get is about half as big as M max. So what that means is the H reflex is activating about half of the motor neurons to that muscle. So Mateos is now going to deliver the stimulation at a few different intensities and see what the largest H reflex we can get is.
Okay, I reduced intensity, stimulated. The H reflex is starting to appear here. I reduced it again, stimulated. It's getting a bit bigger. Reduced again, and reduced again. We went quite high. Reduced again, and you only have an H reflex. There is no M wave here at all. So
that H reflex is about 3 squares, so that's like 30% of M max. And we'll see if we can get it bigger. Oh yeah, there's a big one. So what's that one?
5.
5, so that's more than 50% of my max M wave. It's about the same size. There's a big M and a smaller H.
I'm manipulating stimulation amplitude.
It looks awesome.
It does.
Mateos and I want to collect this data because it looks so good.
We would be hearing laughs if you have microphones. This was about that.
So when we set our stimulation intensity to generate contractions for rehabilitation, we want to have a large H reflex and a small M wave because we want to produce contractions through that reflex pathway through the spinal cord and recruit those fatigue resistant motor units first. And really there's two reasons for that. One is to produce contractions that don't fatigue as much, so that last longer. But also it's those fatigue resistant motor units that tend to die off if they don't get used. So if you don't use them, they tend to die off. So for people with a spinal cord injury, those fatigue resistant motor units die off. So if we can keep them active, it should help to keep the muscle from getting smaller and wasting away. So that's kind of, there's two reasons that we think this is going to be a good idea. One is to help people move, so maybe exercising on a bike or walking to allow those contractions to last for a longer period of time. But the other is to keep the muscle more healthy. So many of you probably know people with a spinal cord injury and know that the muscles tend to just get smaller and smaller. If we can keep those fatigue-resistant motor units active, we think that we can prevent much of that muscle atrophy or muscle wasting from happening, and keep the people more healthy, really is the goal.
They probably would like to hear how does ages change during walking or during standing?
I didn't mention that.
Okay.
So that's pretty much, I think, all we had to show you. So talked a little bit. Oh, do you want to? Can you pull that up? Okay. The last thing we're going to show you then is recruitment curve, and we have a fancy program here that allows us to plot a recruitment curve as we're collecting the data. So that's the last thing we'll do. And Mateos, what he'll do is deliver the stimulation from very low to very high. And hopefully what you'll be able to see on the computer screen is the recruitment curve as it develops. So I'll stop talking and Mateos can show you that development of that recruitment curve.
Sorry, I have to do some preparation. It's fancy, but it's not that fancy.
No. This may or may not work.
I think it will not work somehow.
Is that right?
Yeah. This,
What if you deliver a stim manually? Does it see that?
no, it won't plot it.
Okay.
The software is not plotting. I'll just.
We'll just give this another try for a minute, and if it doesn't work, we're happy to answer some questions. I can't see the chat thing.
There is no questions yet.
Okay.
You guys can ask questions if you want, starting up while we plot this.
Doesn't look as good. I don't think so.
Oh, I see. Should be working.
It was working before we started, of course.
Philip is writing something.
Somehow it's not working.
Okay, we have technical difficulties. We're going to have to... I think we'll stop it there, and we're happy to answer any questions. What does that say?
So thank you guys for this awesome talk and practical lesson about the electro simulation. And people that people participating in the webinar, if they want, they can send questions em português. Se vocês tiverem dúvidas, podem enviar em português, porque nós traduzimos para o Dave, e também o Matheus é brasileiro, então ele pode fazer esse meio campo conosco lá. Não fiquem com vergonha de fazer perguntas.
Coming up? How can you tell?
It shows in the bottom like someone's writing. This was my question. It's all it takes. I see a bottom.
Oh. Thank you.
Do we know? No.
I talked to the postdoc guy yesterday. We're gonna. Oh, I didn't probably tell you about the postdoc guy. Oh, another question. Oh, maybe there. Let me start this guy. Yes, good question. And actually, when I was at the meeting in Brazil, I saw a lot of talk about exercise in the elderly and even using electrical stimulation for the elderly. And yeah, I think it would certainly work in lots of populations. So the idea of producing contractions in a more, using electrical stimulation, but in a more natural way. So when you stimulate motor axons, you activate the axons randomly, so you don't get them in this natural order from fatigue resistant to most fatigable. You just get them randomly. So I think if you can produce contractions through that reflex pathway, it would have advantages really for anybody who's using electrical stimulation for rehab, whether it's to build up the muscle or to produce functional movements. It should be beneficial. Yeah, so the question is, we needed to reach maximal, and we really only do that for experimental reasons, so that we can quantify something in terms of a percent of that maximum. It can hurt. It can hurt, but it's a single pulse, so we're not delivering that maximal stimulation repetitively. So it's not that bad. And with people with a spinal cord injury, especially if the injury is a complete injury, often they have no sensation at all. So for them, we can turn up the stimulation as much as we want, and it doesn't hurt. For other populations, though, sometimes they have heightened sensitivity, or their skin is more sensitive than it is normally, and then the stimulation can be painful, and that is one reason that people don't use it for rehab, is that they can't really get a big enough contraction without it being painful. So a lot of people don't use it because of the pain. Some people use it even though it's painful because it's beneficial. So definitely discomfort is a problem, and it actually something that Mateos is working on for his PhD, is looking at whether or not different ways of stimulating can generate contractions that are cause less pain.
Yeah, because spinal cord injury subjects, they cannot, even though you can stimulate them at a quite high intensity compared to healthy individuals, you can injure them. And when we start to create new protocols of stimulation, these kind of population cannot report how they feel or what the stimulation is causing. So we usually start with healthy subjects, and then we got reports of pain, and then we got reports of force production, so we can use these information to guide stimulation in spinal cord injury population to try to avoid injuries. And the Mmax, the maximal stimulation, that to get that maximal M wave, the biggest one at high stimulation intensities, is the same idea as what people use for interpolated twitch torques. When you do MVCs, maximal voluntary contractions, and then you deliver a twitch on top to see if you are really stimulating everything in that muscle. So this is why we do maximal, this is one of the reasons why people do maximal stimulation. We do it just to normalize our EMG data, to get everything that you can. This is 100%, and then you normalize it by that.
Very good.
Right on.
So, if you have some additional questions, please let me know. Otherwise, I will be happy to thank you, Dave and Matthews, again for the talk and for the time and all the presentation and preparation for this webinar. That was very important for us. And this webinar was recorded, so we plan to put all the webinars from this year on YouTube as soon as possible, so people will be able to join the lectures and learn from you in the future, for those that were not able to be online today.
Sounds great.
Yeah, and I think we don't have any questions anymore, so...
Okay.
Thank you, Dave, for your time. Thank you, Mateus, for everything. And congratulations on your great job. And I hope to see you soon again in Brazil or Canada or another place, I don't know.
I would like that very much.
Okay. Thank you, everybody, for joining us for this webinar. And next one will be in the end of May. With Dr. Daniel Ribeiro from Otago University. And he will be talking about the manual therapy for shoulder injury, so people from physiotherapy, physical education, that will be interested. Keep in mind this date, right?
All right. Thanks a lot, everybody. Bye.
Bye bye.
Yep.
Bye bye.
Helen, nice to hear from you.